So what's occurred in the 25 years since this idea from Dr. Jeff Bland and Dr. David Jones and Dr. Sid Baker and others has emerged as they sit in the front row here? So we have more than 1,000 people here gathered to talk about the omics revolution. But it's really about the healthcare revolution and the revolution of health itself. So that leads me to our first speaker of the morning, Dr. Jeanette McCarthy. And a quote from her in a recent editorial in Genome magazine. She's the editor-in-chief of Genome magazine, a patient-focused magazine, journal, to talk about the genome. And she says, to me, the idea that healthcare consumers can't handle their own health data, especially something as personal as their own genome, is unsettling. Now, that's a startling thing, and as I talked with Dr. McCarthy, she has allowed the environment, the nature of what's happening in the change in healthcare to bathe over her genes and her experience, to create a phase shift in her life, to focus on precision medicine, a new term brought forth by the President and the White House of saying, how do we really focus on individualized and personalized care? How do we bring that forth? And bring it forth in a way, as Leroy Hood from the Institute of Systems Biology would talk about, as participatory, where our patients are engaged in the process and where we're engaged in the process. And as we'll find, in order to do that effectively as providers, we need to be educated on, how do I use these tools? How do I bring this forth? So Dr. McCarthy is an adjunct professor at Duke University School of Medicine in Personalized and Precision Medicine. She's also on the faculty and associate professor of medicine at UCSF, where she's brought forth a course, a huge online course through Coursera, where many thousands of people were able to learn more about precision medicine and about personalized medicine and the genome. So they're gathering that information. She's an educator. She won the Golden Apple Award as an educator from UCSD. And she's brought forth these ideas not only in industry, but in academia, with more than 50 published papers. As I said, she's the editor-in-chief of the magazine Genome. And where she's focusing her work is on, how do we educate and bring these ideas forward, not only to providers as we have in this room, but to our patients, so that they can leverage the data. Knowing the information about what's in my genome will help me to be able to make choices that will allow me to live longer and with less suffering. And that's where we're going here at the Institute for Functional Medicine. We look with great joy, we look forward to bringing this course forward over the weekend, over the next 3 days. And it's with great honor and respect that I introduce Dr. Jeanette McCarthy.
Hi, thank you. Thanks. Good morning. Well, it's a real pleasure to be here today, and I'm really excited that the Institute decided to have the omics revolution as the topic of this year's conference. You see, I'm an omics researcher by training, a genetic epidemiologist, and I spent most of my career in industry and academia researching the genetic basis of complex diseases. But about 4 years ago, I decided to take a shift in my career and focus exclusively on educating stakeholders in this exciting field of precision medicine. Precision medicine, as you know, is using molecular information about an individual to tailor treatment and healthcare decisions. This isn't a new idea, although the term precision medicine is relatively new. The concept has been around for a long time. But it's really in the last couple of years that things have really taken off, As you heard President Obama mention precision medicine in the State of the Union earlier this year in the context of the precision medicine initiative that the government started. Today, I would like to teach you about some of the current and future applications of precision medicine in the clinic. I'd like to tell you a little bit about some of the hurdles that we face trying to implement precision medicine. But before we do that, I'd like to take a little bit of time to give you a basic primer on genomics. You see, I realize that many of you may never have had a genetics class in your life, and even if you did, it was probably 5, 10, 15, maybe 20 years ago. And although the basic principles of genetics haven't changed, the breadth and the depth of the knowledge that we've gained since the sequencing of the human genome is noteworthy and worthy of review. So I'd like to start kind of basic, but move very quickly. First of all, talking about DNA and the human genome. So DNA, very basically, is a polymer of repeating units called nucleotides. Nucleotides are sugar, a phosphate, and one of 4 nitrogen bases that we abbreviate by their single letter codes, ATGC. And in its naturally occurring state, DNA is not a single-stranded molecule, but rather a double-stranded molecule. These 2 molecules join together in a specific way to give you the classical double helix shape of DNA. When we talk about a DNA sequence, we're really talking about understanding the order and composition of these nucleotides in the genome. What I have shown here is a stretch of 34 nucleotides, or base pairs, as we like to call the unit of measure. To put this into context, the human genome consists of 3.2 billion nucleotides, or base pairs. So it's quite large. And if you think about the DNA in a human genome, it doesn't exist as one long molecule, but rather it's broken into 23 discrete units that we call chromosomes. These chromosomes are located in the nucleus of every cell in your body that has a nucleus, and that DNA is, for the most part, identical. Since humans are diploid organisms, we have, in fact, 2 genomes, one that we've inherited from our mother and one from our father, so we, in fact, have 6.4 billion nucleotides of DNA, or 46 chromosomes. So why is DNA important? Well, DNA is, first of all, the unit of heredity. It's what's passed down from generation to generation. But more importantly, DNA provides the instructions from which all of the proteins in your body are made. And the segments of DNA that code for proteins are called genes. There are approximately 23,000 genes in the human genome, but this only comprises less than 2% of all of the DNA in your genome. So the big question everyone's asking is, what is that 98% DNA that's not coding for genes doing? And the short answer is, we don't really know. There have been estimates in the last few years about how much of that DNA is actually functional versus what we like to call junk DNA. And those estimates range from 8 to 88%. So we're again not quite sure exactly what all of that extra DNA is doing. There's some thought that it is involved in regulation of gene expression. There's some thought that it's involved with folding of DNA in chromosomes, but the ENCODE project is a big project ongoing right now to try and figure out what exactly that DNA is all about. But in the meantime, we're happy to focus on this 2% of the genome that codes for genes. So the process by which DNA codes for genes is protein synthesis, where DNA is transcribed into an intermediate molecule, RNA, which is then translated into proteins. Human beings are 99.9% identical. But what that means is at 0.1% of your genome, you differ from somebody else. And that equates to about 3 to 4 million nucleotide positions. Now, mutations are a naturally occurring phenomenon. Every time your cells in your body replicate, the DNA needs to replicate and divide, and during that process, some mistakes are made. And fortunately, we have enzymes that repair these mistakes, but those enzymes aren't perfect. And so once in a while, a mutation sneaks through, and it persists. Now, if that happens in an individual cell in your body, it's typically of no real consequence. There are some exceptions that I'll talk about in cancer, but for the most part, a single mutation that occurs in a single cell in your body doesn't really do anything. But if you have a mutation that happens to occur in the process of meiosis, the formation of the gametes, those mutations can actually be passed down to any offspring that come from those sperm or egg, and those mutations will appear in every cell in the body of that individual. Now, you notice I have mutation/variation. I prefer the word variation because mutation to me has a really bad connotation. When we think of mutations, we think of bad things, but in fact, mutations are random neutral events that occur, and this leads to variation in the genome. Now, we know that there are a number of contributing factors to the rate with which mutations occur. We know that cigarette smoke, UV radiation, X-rays all induce DNA damage and cause mutations. But one new interesting factor that contributes to variation is paternal age. As women, we're used to hearing about advanced maternal age and the dangers of mutations if you wait too long to have children. But a study a few years ago found that, in fact, males are contributing an awful lot of genetic variation into the population. It turns out that every newborn has about 50 to 100 new mutations that have arisen just by chance out of this process of DNA replication. These mutations are actually 3 to 4 times more common coming from males versus females, and paternal age is a big factor in this. With every year of advanced paternal age, approximately 2 additional mutations can be found in the offspring of that person. So I don't know if this is a good thing or a bad thing. Part of me thinks, well, maybe we can blame advanced paternal age on all of these rare genetic diseases that we get. But on the other hand, I think, well, not all mutation's bad. In fact, mutation is what drives evolution. So maybe older males are propelling the species forward through evolution. I don't know. But in any case, we know that paternal age is also a factor in increasing mutation rates. The different types of variation that exist in the genome, I like to think about as large and small. The large variants include things like chromosomal trisomies and monosomies, these numerical aberrations, where entire chromosomes are either found in excess, usually 3 copies, or perhaps just 1 copy. Other large types of genetic variation are structural variants. This is where entire chunks of chromosomes, for example, are moved from one location to another in the genome, such as in translocations. These types of variation are very rare. And the other thing is that they're very big, so when they do happen, they're very deleterious. The more common type of variation that you see in a human genome comes in a couple of different forms. One form, which we've learned a lot about in the last few years, are these copy number variants. This is where stretches of DNA, and these are not small segments, they can be a few kilobases in length or larger. They're either deleted from the genome or duplicated in tandem, once, twice, 3 times. And if these occur in gene regions, they can also be deleterious. Every person has about 800 of these in their genome. The more common type of variation, though, involves single or 1 or 2 nucleotides of DNA. These single nucleotide variants are exceedingly common in the genome. Every person has about 3 to 4 million of these. So how do we know so much about human genome variation? Well, it's through sequencing of the human genome. We know that as we've progressed technologically, that the cost of genome sequencing has come down, as well as the time it takes to do it. When the first human genome was sequenced, it cost about $3 billion for that genome. Today, it costs about $1,000. And more people are getting their genome sequenced. Some people estimate that within the next 10 to 20 years, whole genome sequencing will be a routine part of medical care. every person will have their genome sequenced. But today, only about 230,000 people have their whole genome sequenced, including a few of you in this room, I suspect. But that's a very small number of people compared to the population of the US and indeed the population of the world. Now, not all variation is deleterious. First of all, most of the variation in our genome occurs in non-gene regions. So, to the best of our knowledge, that's having no real impact on health. And even those variations that occur within the coding regions or the genes may or may not be deleterious. Some variants result in truncations of the resulting protein that gene codes for. Some variants result in single amino acid changes, which may or may not be deleterious. And other variants have no real effect on the protein function or structure. What we're really interested in, though, is how much of this variation is linked to disease. And a lot of research has been done in this area, not just since we've been able to sequence human genomes, but for many, many years. And what do we know? Well, there are really two types of variants that we see. There are variants that have very large effects, and there are variants that have small effects. Variants that have large effects, these are usually very rare. They tend to be linked to these Mendelian diseases. These are diseases where if you have one of these mutations, you have a very, very high likelihood of getting the disease. So we call them highly penetrant. And a lot of times these are de novo. They're brand new. And sometimes they cause very serious abnormalities so that that individual would never reproduce and pass that variant down. These types of variants are very useful clinically because if you can test for them, you can predict with pretty good accuracy whether someone will get a disease or not. The other type of variant and disease is a variant with a small effect. Unfortunately, most of the diseases that we're interested in, most of the diseases that affect people are of this type. These are common, complex diseases, where these diseases are due to a combination of genes and environment. In fact, there are some forms of the disease that are purely genetic, perhaps, some that are purely environmental, but many that are really due to the combined effects of genes and environments. So what do we know about the genetic basis of these diseases? Well, any of the gene variants that we found to date really have a very tiny, tiny effect on disease, on the order of 1.1 to 1.2-fold increased risk. From a clinical standpoint, these aren't very useful. Now, I spent a lot of time talking about DNA because, in fact, that's the basic molecule and the molecule from which we know the most information. And why is that? Well, it turns out that DNA is relatively easy to study. When we talk about genomics, technically that includes the study of DNA as well as transcriptomics or RNA. The difference being every cell in your body has virtually identical DNA, but the transcripts that lead to the proteins are going to differ from one cell to the next. And as you move to the right in this diagram here, you get closer and closer, going from mRNA to protein to metabolites, closer and closer to the biochemical and clinical phenotypes that we care about, those related to disease. So ideally, we really want to be shifting our study to the right on this graph and studying these things that are much closer to disease. But it's not that easy to do. DNA is relatively easy to study because, number one, it's relatively stable. If you think about over time, or even within the context of one day, from morning to evening, or from today and 5 years from now, you think about the proteins that are expressed in a different cell, they're going to change. They're going to vary over time. They're going to vary with environmental exposure. They're going to vary with different developmental stages of an individual. So those are not very stable. DNA, my DNA today, is going to be the same as my DNA 10 years from now. It's going to be the same whether I smoke or whether I don't smoke. So DNA is relatively stable. That's one of the reasons we spend so much time studying it. The other aspect is that it's very accessible. If I want to study diabetes, and I want to look at the proteome and understand what are the different proteins involved in diabetes, I would probably want to look in, say, a pancreatic beta cell or some tissue that's really relevant to that disease, and I would want to look at the proteins in that tissue. But those aren't always accessible to me. I can't always take that biopsy. I can't always get that information. If I want to study the genetics, if I want to study the DNA as it relates to diabetes, I take a sample of blood. The DNA in my lymphocytes is the same as the DNA in my pancreatic beta cells. There's no difference. And so it's really the stability and the accessibility which lead us to studying DNA. It's just easier right now. But we recognize that we need to move further to the right, and indeed we've made progress in this area as well. Last year, Nature published the first draft of the human proteome, and I know you'll hear a lot about the human proteome today and throughout the rest of this conference. But basically, the goal of any of these big projects is really to, first of all, map out the landscape of, in this case, the proteome in healthy individuals. And that's exactly what they did. What did we find? Well, we found that different tissues in your body have different protein profiles. Okay, no surprise. We also found that over time, for example, comparing fetal to adult liver cells, you see different proteins expressed. Again, no surprise. So again, we now have this blueprint for the healthy human proteome. The next step is to try and link this to disease. How does the proteome change when a person goes from health to disease? Similarly, we're beginning to make some progress in the metabolome. This is the full complement of small molecules that are the result of metabolism in the cell. I think the metabolome is going to be very interesting for common complex diseases. These are the diseases where we have many different genes, many different genetic variants leading to disease, but each with a very small effect, which again aren't very useful clinically. But if you think that, well, maybe these individual genes represented by the different colored dots on this top graph, at some point they converge on a common metabolic pathway. And then maybe by being able to study that metabolic difference, we'll be able to more accurately predict disease. A number of studies have been done recently looking at the link between genes and metabolic profiles. The bottom graph shows a composite of several genome-wide association studies that were done, looking at 163 metabolic traits and finding significant associations with specific gene variants and each of those traits. So we're making headway there. I know there's a group at Duke who's also looking at metabolic profiles. So, for example, not just a single metabolite, but groups of metabolites and how they link to genetics, and especially in the area of cardiovascular disease. So I think there's a lot of promise in using these other omics tools as well. The final frontier, probably the most exciting thing going on right now, in my opinion, is the human microbiome. So trying to understand these companion microbes that outnumber our human cells 10 to 1, what are they doing and how are they affecting health? The Human Microbiome Project is surveying the landscape of 300 healthy people at 18 different body sites to try and understand what microbes are living there. And people are starting to put this together with respect to health and disease. Again, you'll hear about this more in the conference this week. But one interesting study that I wanted to highlight is one linking diet, microbiome, and disease. And this study looked at dietary phosphatidylcholine and the effect that intestinal microbiota have in metabolizing this, and the byproduct, the LMAO, increasing risk of cardiovascular disease and atherosclerosis. So really exciting things linking diet, microbiome, and health. So next I want to turn our attention to applications of precision medicine in the clinic. What is actually going on today and what are we excited about? We know genomics, and in particular genetic testing, happens across the lifespan. And there's a general trend to get tested earlier and earlier. And what this allows us to do is to move from a state of diagnosing disease to screening and ultimately preventing disease. So we have testing in adults, we have testing in children, we move it to newborn, prenatal, and even preconception. Preconception carrier screening, many of you may have already undergone this yourselves in the context of family planning. Basically, this is a screen for inherited recessive diseases. These are diseases where you require mutations coming from both sides of your family in the same gene in order to get the disease. This type of testing is really looking for people who carry a single copy, and if both parents carry that single copy and have children, there's a 1 in 4 chance of them getting this recessive disease. It's estimated that 1 in 4 people are carriers for at least one recessive disorder. Typically, this type of screening has been conducted using ethnicity as a cue. So, for example, when I was pregnant, I was tested for cystic fibrosis because that happens to be the most common recessive genetic disease in European populations. If you're African American, you may have been offered sickle cell testing because that is very common in African American populations. And if you're of Ashkenazi Jewish descent, you may have been tested for Tay-Sachs disease. So maybe 1, maybe 2 genetic tests that have been available over the years. But now, with genome sequencing and all of the genes we've discovered for recessive traits, there are companies offering screening for over 100 disorders. You can go to Council Genomics or Good Start Genetics, and you can get a panel for your patients and test them for all of these genetic disorders now, before they have children. Another exciting area is in prenatal diagnosis, moving up the spectrum from preconception to now when you're pregnant, carrying a child, and you want to screen them for a genetic disorder. Historically, women have undergone prenatal screening using blood tests. They've also, in recent years, been using high-resolution ultrasound to try and detect chromosomal abnormalities. And what was tested for basically are trisomies, so some of the gross chromosomal abnormalities, as well as neural tube defects. Women who had a positive screening test would be offered amniocentesis or chorionic villus sampling, where they would actually invasively go in and get a sample of the fetus to test to confirm that they had one of these diagnoses. This invasive procedure poses a risk to pregnant women, which is not insignificant. So it was really exciting a few years ago when we were able to begin to detect fetal DNA in the maternal blood. It turns out that as the fetal cells are dying, they enter the maternal circulation, and beginning at about 6 to 8 weeks gestation, about 10% of the DNA in the maternal bloodstream is actually from the fetus, and we can detect that. With next-generation sequencing now, we've been able to take a blood sample from the mother and detect the fetal DNA and determine whether that fetus is actually carrying 3 copies of chromosome 21, 3 copies of chromosome 18, any of the typical trisomies or monosomies that we would screen for usually using the typical screening and amniocentesis follow-up. This is really exciting. In fact, last week or the week before, another landmark paper was published which found that not only is this a good test for screening, because as we started to introduce this technology, we were first using it as a screening modality, following it up with amnio still. But now this study came out and showed that it's even better than amniocentesis. And so we'll see a big shift now, moving away from doing these invasive amnios to simply using this non-invasive prenatal testing in pregnant women to diagnose genetic disorders in their children. And for now, the technology is really limited to very large genetic changes, these chromosome monosomies and trisomies, but slowly it's moving towards smaller and smaller variation. Some of these tests offer testing for structural variants, these large duplications or deletions of entire chunks of chromosome. And as we push this technology further, we'll begin to see our ability to detect these smaller and smaller variants. One of the most exciting applications of genomic medicine right now, I think, is in diagnostic sequencing for rare idiopathic disorders. Rare diseases, by definition, affect 1 in 200,000 people. These are exceedingly rare, things you've probably never heard of. I have examples of some listed on this slide. I don't know anybody who has had one of these, but because there are about 7,000 of these rare diseases, collectively they're actually quite common. It's estimated that 1 in 10 Americans is affected by a rare disease. Most of these affect children, and it turns out that about 80% of these are genetic. We know the genetic basis of about half of these, but for the other half, we haven't found the genes yet. Many of these diseases present as diagnostic dilemmas. A typical diagnostic dilemma begins with a child coming to their doctor with some strange array of symptoms. They get tested, they get diagnosed. Happens to be a misdiagnosis, but they don't know at the time. They undergo treatment, which is ineffective. In the meantime, their disease progresses. They have more testing. Maybe they get a new diagnosis, more ineffective treatment, and so on. Around and around it goes. The typical diagnostic odyssey lasts maybe about 7 years or so before they finally get a diagnosis. That's why it was so exciting in 2006 when the story of Nick Volker broke. Nick was a little boy who at 2 years old began to exhibit some intestinal issues. He had some serious issues with his gut. He, at 4 years old, I think it was, weighed 17 pounds, had to have part of his colon removed. You can see this picture of him, not a happy boy. He had 160 surgeries before being referred to Howard Jacob at the Medical College of Wisconsin. Howard was the first person who successfully carried out whole genome sequencing on a patient and diagnosed their disease. Through Howard's work, they found a mutation in a gene called XIAP, which led to a diagnosis of a very rare form of inflammatory bowel disease in Nick, which could be treated with a cord blood transplant. He was treated, and look at him. So this is really exciting. I think this really paved the way for more studies like this. And since 2006, you can see this growth in the number of children whose diseases have been diagnosed using whole genome sequencing. Now, I don't want to set your expectations too high. This doesn't work in everybody. This success rate is about 25 to 30%. And even if you have a diagnosis, it doesn't mean that there's a cure. But for many parents, that doesn't matter. Having an answer is enough. As we move into adulthood, we start thinking about the genomics of complex diseases. Complex diseases, again, due to a combination of genes and environment, very difficult to disentangle the two. Breast cancer is a great example of a complex trait because there are forms of breast cancer that are essentially very much driven by inherited genes, other forms that are driven purely by environment, and then, of course, a bunch that are the effects of both genes and environment. It's estimated that about 10% of breast cancer is hereditary. And what do we know about the genes underlying this hereditary breast cancer? Well, we only know about half of the genes. The other half of hereditary breast cancer remains unexplained. And among the genes that have been discovered, of course, the two most popular ones, BRCA1 and BRCA2, account for about 50% of inherited breast cancer. There are other genes, for example, P53, P10, ATM, and such, which underlie rare inherited cancer genetic syndromes for which breast cancer is one of the features. And then you have a bunch of these common variants that I talked about. I don't know what the latest count is, but at least 79 different genes where common variants contribute a very, very small amount of increased risk in people who carry them. So these high penetrance variants, the ones for which if you carry this mutation, you have a very high risk of developing cancer, are very useful clinically. In fact, I'm sure you're all aware, not just because of Angelina Jolie, but maybe your own experience with patients, that there's testing available for hereditary breast cancer. Dozens of labs offer this test. It used to be that everybody went to Myriad Genetics, but since the Supreme Court ruled that DNA was not patentable, that opened up the field so that other companies could offer these tests. And indeed, a bunch of people jumped in. A number of companies offer tests not just for BRCA1 and BRCA2, but panels of genes. And one company that just came to my attention just announced it's opened a few months ago, I think it was, is Color Genomics. This is something that's really exciting because most of these genetic testing companies offer BRCA or breast cancer screening tests for around $1,500. Color Genomics offers this test now, a panel of 17 genes for $249. And it's really exciting. I mean, this is something that is just really revolutionary and democratizes genetic testing. Breast cancer and the BRCA genes in particular get a lot of attention, but you can get tested for a number of other disorders as well. Lynch syndrome, an inherited form of colorectal cancer, is also a test that's available, as well as some other rare cancer syndromes. In the area of cardiology, there are very good genetic tests for cardiomyopathies, long QT syndrome, things like that. There are also genetic tests available for some Mendelian forms of epilepsies, autism, obesity, diabetes, etc. But again, these are for these Mendelian rare forms, highly penetrant forms of the disease. What about common complex testing? Well, many of you may have done 23andMe testing, had your own genomes, genome-wide SNPs tested, as I did. And if you get results for their risk profiles for different diseases like breast cancer, these results are not often very useful. For example, based on 7 different genes, each having a very, very tiny effect, if you put all that together, it turns out that I have a 13% risk of breast cancer. But compared to an average population of 14%, that really doesn't give me much. So I think we have a long way to go in the area of complex diseases and these variants that have small effects and being able to use these in a clinical setting. Another area that you may have heard of is pharmacogenomics. I know you'll have some additional talks on that during this conference. Basically, pharmacogenomics is using your genetic information to help tailor your treatment. There are 3 different areas that this is useful in. There's efficacy-based pharmacogenomic testing. A great example of this is with ivacaftor for cystic fibrosis. This is a drug that was developed specifically to target certain mutations in the CFTR gene. Mutations in CFTR, of course, cause cystic fibrosis, but there are over 1,000 different mutations that exist. This drug only works if you have one of like 7 or 8 different types of mutations that have a specific effect on that on that chloride channel. So efficacy-based testing, in order for this drug to work, you need to have genetic testing to determine if you have one of these variants. And only in that case will this drug work. That's a useful pharmacogenomic test. There are toxicity-based pharmacogenomic tests. These are tests where genetic testing can help you avoid a rare severe adverse event. A great example here is with abacavir, which is used to treat HIV. HIV is the target of this drug, but the drug also happens to co-react with your human leukocyte antigens. In particular, if you have HLA-B5701, this drug will bind to it and in some cases cause this severe hypersensitivity reaction. If you have any other HLA type, you're fine. So drug labels for abacavir include genetic testing as part of the indications for use. The other area that pharmacogenomics is used in is in determining the dose of a drug. This is particularly important for drugs that have a narrow therapeutic index where you really need to get the dosing right. An example here is with warfarin. Warfarin is metabolized by CYP2C9, and if you happen to be one of the people who is a poor metabolizer based on your genotype at your CYP2C9 gene, you won't be able to metabolize this well, and the result is that in your bloodstream, you will have more drug than you thought you had. This can lead to toxic side effects, such as brain hemorrhage. And so in cases like this, you may want to have genetic testing in order to help dose. There are a number of different pharmacogenomic tests out there. There are 158 drugs that have pharmacogenomic information in the label. Now, I have to tell you, not all of these are useful markers. One thing that surprised me is the fact that just because something appears in an FDA drug label and says there's a pharmacogenomic test for this drug, doesn't mean it's a good test. And it's very difficult to know which ones are good and which ones are bad just by reading the label. Most of the pharmacogenomic markers are in the area of oncology. These tend to be very useful because these are ones, in fact, where we're looking at tumor markers and targeting therapy. They're efficacy-based pharmacogenomic markers. Drugs were developed specifically for people with these mutations in their tumors. So oncology is a big area. But you see pharmacogenomic markers across different therapeutic areas. The area that's probably the most useful in the area of precision medicine right now is in targeted cancer treatments. So cancer is always a genetic disease, and that confuses people sometimes because they think genetic means hereditary, but that's not what I mean. I mean cancer is a genetic disease because it affects the DNA in your cell, and as opposed to inherited mutations, which appear in every cell in your body, the types of mutations that are most relevant in cancer are somatic mutations. These are the mutations that affect an individual cell. Maybe it's a breast cell, which could potentially lead to breast cancer. Cancer is a multi-stage process where you're basically bypassing growth control. So every time your cells divide, there are instructions, growth factors that either promote or inhibit this cellular proliferation. And there are molecules that control this, cellular receptors, growth factors, growth inhibitors. And what happens is this whole process goes awry because of the accumulation of mutations, sometimes in this very machinery, sometimes further downstream. A cancer cell has acquired a number of different mutations, which eventually lead to the development of cancer and the loss of growth control. The tumor proliferates, invades the normal tissue, metastasizes, and is typically diagnosed only when it metastasizes. The tumor, as it continues to grow, continues to acquire mutations. With our ability to sequence human genomes, a lot of work has been done now to try and understand what are all of these mutations that occur in these cancer cells, and which of them are important? Which of them are the ones leading to this uncontrolled growth? So a number of different tumors have been sequenced. For any given cancer, sequencing of hundreds of different tumors have led to our understanding of this mutational burden, especially in solid tumors. As you see in this graph, there are many different types of variation that occur in tumors. Most of them are, of course, these single base substitutions. But you also have insertions, deletions, amplifications of these genes, whole gene deletions, translocations. Basically, these tumor cells at the genetic level are really messed up. And the interesting thing is, if you look across different tumor types, you see different mutational burdens. On the right-hand side, you see glioblastomas, medulloblastomas, cancers that occur in childhood, where these children haven't been alive a long time, and so their mutational burden is quite low. You move to adulthood, there's been more time to accumulate mutations in these tumors. You have some special cases like colorectal cancer. Why would colorectal cancer be so high? Well, this is principally due to one inherited form of colorectal cancer, Lynch syndrome, which happens to be due to mutations in that very machinery that's supposed to be correcting all of the mutations that occur during cell division. So your mismatch repair enzymes are supposed to go in and take care of variants, but they don't do their job, and so you get an accumulation. What's not on here are other cancers, lung cancer, for example, melanoma, due to environmental exposures. Those also have very high mutation burdens. But it's not good enough to just know how many mutations these cancers have, but what genes are mutated, and which among those are the ones driving the cancer forward. Well, we've begun to make progress in this area. Here's an example of a chart that shows frequently mutated genes in non-small cell lung carcinoma. And it's really interesting as I look at this pie chart, it's amazing how we're redefining cancer. This is no longer non-small cell lung carcinoma. This is ALK positive lung cancer. It's FGFR positive lung cancer. And so you begin to really look at the molecular level of these tumors, and we've been able to take advantage of this then to develop targeted therapies. If my tumor happens to have EGFR mutated, I can take a drug now, cetuximab, that targets specifically that tumor protein. In non-small lung carcinoma, we have 3 different drugs now available that target EGFR. There's another targeted therapy available for people who have the ALK protein expressed. And there are a number of drugs in clinical trials that are targeting some of these other proteins. These drugs are only efficacious in people who have these specific tumor markers. And so typically, again, you would order a companion diagnostic genetic test to determine what their tumor profile is, and based on that, you could prescribe some of these drugs. Now, one interesting thing is, as you begin to look across different cancers, and in this graph we have 9 different cancers shown, and a number of different tumor markers, you can see that for some tumor markers, they're common across multiple different cancers. The BRAF V600E mutation, a single amino acid change, is found in 60% of melanomas. It's found in 100% of hairy cell leukemias. It's also found in 2% of non-small cell lung carcinoma patients. Now, there's a targeted treatment available in melanoma for people with this mutation, and it turns out that some case studies have shown that non-small cell lung carcinoma patients who also have this BRAF mutation are responding to this drug. So now we're beginning to see some off-label use of these targeted therapies based on molecular profiling. So it's really exciting. This doesn't always work. Sometimes it does, sometimes it doesn't. Well, more research needs to be done to figure out in which cases this is effective or not. But it's an option for patients. This figure shows that for some cancers, a large percent of the patients are actually candidates for targeted therapies. Melanoma tops the list. 73% of melanoma patients are estimated to have a target that is amenable to this targeted treatment. So it's very exciting to see, as the field progresses, more and more patients being candidates for these targeted therapies. So those are the main clinical applications that are available today. These are things that are out there today. Next, I would like to turn our attention to some of the hurdles that we face in implementing or practicing precision medicine. So, healthcare providers are really the gatekeepers to precision medicine. And I like to characterize healthcare providers with respect to precision medicine using these four traits. First of all, healthcare providers are unaware of some of the advances in precision medicine. Healthcare providers are skeptical about the validity and utility of these tests. Healthcare providers are unsure of themselves. They're not confident in their own understanding of genetics or these tests, and therefore afraid to use them. And healthcare providers are cost conscious. I launched a MOOC, as you mentioned, through Coursera a couple of years ago. This was to teach healthcare providers all about precision medicine. And before I launched this course, I did a survey. I did a survey of my colleagues at UCSF, a formal survey, right? Went out to everybody asking, are you interested in a course like this? And this is one of the responses I got. It's very unclear to me this should even be in the top 25 topics in primary care. I see the value in oncology in advancing knowledge, but what else? Hence, healthcare providers are unaware. Did you not know about non-invasive prenatal testing? Did you not know about pharmacogenomics? Did you not know about all of these things that are going on out there? And I think the answer is no. Healthcare providers are skeptical. I don't and shouldn't adopt any new technology, test, or therapy until patient-centered improvements and outcomes are demonstrated, and until that randomized controlled trial data is available, I'm going to tune this out. We are driven by evidence-based medicine, and I want that evidence. I want those guidelines. Show me the guidelines, and I will practice this. And, to an extent, that's a very valid point. But when you think about precision medicine, what are we doing? We're taking a disease like non-small cell lung carcinoma, and we're dividing it into much smaller diseases. I'm not a lung cancer patient. I'm an ALK positive. I'm one of those 5% of lung cancer patients who has ALK protein on my tumor. I'm a candidate for this therapy. Do a clinical trial, provide evidence for me. So now we're subdividing diseases into these very small bins and expecting to be able to generate randomized clinical data on these. It's just not going to happen. And so there needs to be a shift in the way we think about evidence. I read a recent commentary in, I think, Nature or Science by Nick Shork that talked about N of 1 trials. How are we going to accommodate evidence, the accumulation of evidence in the context of single patients? And I encourage you to look into that and challenge you how to think about that. And it's not just information, it's not just evidence about the validity of the test, but it's about the utility. Does it save money? Does it improve health outcomes? And if we have to wait for all of those studies to be done before we implement this, we're not going to be moving forward very quickly. Healthcare providers are unsure. This was a survey done by the American College of Physicians that asked internists about their own understanding and confidence in having adequate knowledge in these key areas related to precision medicine. And although 60% said they had enough genetics, they were confident in that, less than 25% thought they had enough knowledge about indications for testing, interpreting results, all of these important things. So healthcare providers are unsure and need to be educated in this area. And finally, something that surprised me was how cost-conscious healthcare providers are. This was another survey I did of the students who were in my Coursera course and asked them, what do you see as the major barriers to obtaining genomic testing for your patients? And the top response was cost and insurance coverage. For me, that was really surprising that my physician would be thinking about that. And it is an important issue. And when you think about insurance coverage, This leads back to this evidence base. Without an evidence base, insurance companies aren't going to be covering these tests. And so we have a real issue now with these precision medicines not being covered by insurance. And hopefully that's something that will change in the next few years. So regulation, I don't necessarily see as a hurdle to implementing precision medicine, but did want to mention it because I think it's an important thing to consider. When you look at the regulatory landscape for omics tests, you have 3 different branches looking at this. You've got the Federal Trade Commission. This is really the group that's looking at false and misleading advertising. Then you have the FDA. Now, it turns out the FDA only regulates devices, which would be test kits in the context of precision medicine. But it turns out that most precision medicine tests, most of these omics tests, are being done as laboratory-developed tests, which aren't under the jurisdiction of the FDA, but rather under CMS. Now, all of that's changing, and you've probably heard in the news that the FDA said that they want to begin to regulate these laboratory-developed tests. So we're going to see a big shift in this space right now. It turns out that the way that they'll be regulating these tests is based on perceived risk. I don't know who's perceived risk, risk of these genetic tests to the patients. But this is really a very topical issue right now and something to keep your eye on. I have confidence that we'll be able to regulate these in a way that doesn't impede innovation or growth in this field. Wanted to spend a couple minutes talking about exciting things on the horizon. Technologically speaking, this is one of the coolest things that is on the horizon right now, this idea of a liquid biopsy. Just as you saw with the non-invasive prenatal testing, where we were able to take a blood sample from a pregnant woman and detect fetal DNA, similarly, we're able to take a blood sample from a patient and in some cases detect cell-free tumor DNA. Now, this is not something that is necessarily in the future because this is being used today in certain contexts. These are called liquid biopsies, these non-invasive cancer tests. They're primarily used today in the context of monitoring recurrence or resistance of the cancer. We're slowly moving to being able to use these liquid biopsies to do the actual tumor profiling to be able to target treatments. And these are tests that are available, from a blood sample, but there are also companies working on detecting these in urine samples as well. But really the holy grail will be when we're able to take a sample of blood as a screening test and diagnose a cancer for the first time. So you think this is really far off in the future, but I want to tell you a story about some news that broke earlier this year. Oops. I had a nice picture there of a pregnant woman, but there was a woman who went in for prenatal testing, non-invasive prenatal testing. And the company that did this test, Genome, contacted her. The medical director contacted her and said, look, I tested your blood, and not only does your fetus look like it has trisomy 13, but it has trisomy 19, and it is missing a chunk of this chromosome, and basically it should be dead. So there's something going on. And it turns out that woman had cancer, and they detected all of these chromosomal abnormalities in her blood sample and diagnosed her with cancer. And it turns out that they had about a dozen patients who had gone through non-invasive prenatal testing and ended up having a cancer diagnosis. So this is coming, and it's really exciting. Another area, again, I put in the future, although it's beginning to happen now, the microbiome. We are understanding the role of microbes in disease, C. diff infection, inflammatory bowel, cardiovascular disease, and so on. And there are some really cool treatment options available. Fecal transplants for C. diff infection, very effective. Custom probiotics. So there's a company, which I can't remember the name now, who is working on genetic testing to profile your microbiome and then offer you custom probiotics based on what's missing or what might be in excess. Dietary modulation, right? The more we understand about the link between diet, microbiome, and disease, we'll be able to offer dietary modulation as an option for therapy. And then there's this idea of creating synthetic microbes, so engineering microbes to actually combat disease. So I think this is a really, truly exciting area. The last area, which is not a technology, but Patrick had alluded to it in his talk, is this idea of participatory medicine. The idea that patients are partners in their healthcare. And it all sounds good on paper, but then I know the physicians in the room are asking, well, how come nobody's accessing the patient portal if they're so interested in being a part of their healthcare? But you do see a subset of patients, at least, who are connecting with each other through sites like 23andMe. They're sharing details about their disease with complete strangers because they want to be part of a group. They want to be around other other patients who have the same disease. They're collecting health information. They're tracking their steps. They're collecting weight, diet information. They're keeping track of this and with Apple Health Watch and Apple iHealth and all of these apps. This is really becoming trendy right now. It'll be great when we're able to share that with our doctor. With a click of a button, just upload all of that information and it goes to your medical chart. Patients are also becoming advocates for their health. This is especially prevalent in the rare disease community, where you have children with these very serious rare diseases. Their parents are becoming extraordinary advocates. There are a couple of organizations like Global Genes, Rare Genomics Institute, and of course Genetic Alliance, where these advocates, these patient advocates are getting together and collectively learning how to lobby for funding, lobby pharmaceutical companies to make drugs for their rare disorder, and so on. So you do see a groundswell of patients who are really interested in being involved, sometimes out of desperation, but nonetheless being involved. And patients are hungry for information. I was sitting in the airport yesterday working on my talk, and I had my screen open, and I had some random older Indian guy walk up to me, and he hunched down and whispered very quietly because his wife was sitting behind me and he didn't want her to hear. And he handed me a piece of paper with his email address on there. And he said, "I want a copy of that," because he saw my slides and he saw cancer on there. He said, my daughter has cancer. Can you send me that? I want cancer information. And I thought, wow, patients are really, really hungry for information. And so, well, I didn't send him this, but I sent him my Coursera course, which has a whole module on cancer. But that kind of leads into this segue for Genome magazine. As Patrick mentioned, this is a magazine that I became involved with a little over a year ago. I can't claim that it was my idea, unfortunately. It was instead Susan McClure, my publisher. She used to publish Cure magazine, which is a patient-facing magazine for cancer patients. And she had this idea about bringing precision medicine to patients. And so this is a patient-facing magazine. It's written for a lay audience. The magazine is free. It's published quarterly, so you can get a hard copy published quarterly. There's also an online version. We have a circulation of about 325,000 right now. What we do is we deliver it to the point of care so that when patients come into their doctor's office, instead of flipping through People magazine or whatever's in there, they can open up Genome and actually read about their health. They can then go home and they can subscribe and have a version, have an issue sent to their house. They can get it delivered every quarter. And what's interesting is that half of our subscription requests are not coming from patients, but from healthcare providers. So patients are hungry for information. Healthcare providers are hungry for information. So I'm really excited about the magazine. There's copies available for all of you here. Feel free to subscribe when you go home. And I want to end with one brief anecdote, and this is a story about a friend of mine. So I'm on Facebook. Many of you are probably on Facebook, and I have friends that I haven't seen in like 30 years, but you get to see what's going on in their life. And one of my friends posted several months ago that he had cancer. He had a rare cancer of his tonsil. And so I watched over the course of the next few months how he wanted to try all of these alternate therapies. He tried dietary therapies and some other things, and really was resistant to the whole chemoradiation thing. And ultimately, a couple months, month or two ago, ended up in the hospital because his cancer, of course, was growing, and it was impeding his breathing and his swallowing, and so he had to go into the hospital and was faced with, well, it's chemoradiation or nothing. And so I'm watching this from a distance and thinking, gosh, I know all of this precision medicine, all of these targeted therapies, tumor profiling, all of this stuff going on. Why can't I help him? And I thought about it, and I thought, well, he should have his tumor profiled, right? He's a perfect candidate. People with rare cancers, have your tumor profiled, and let's see if there are any targets on there that are amenable to therapy, either off-label, for some therapy that's out there, or a clinical trial or something. And so I thought, yeah, that's great. Oh, but it turns out they couldn't remove his tumor because it was too close to an artery. And so I thought, well, that's okay, because we have liquid biopsies, right? We can just take a sample of his blood and detect the cancer cells there and look for tumor profile, and then see what happens, right? It's worth a chance. And then I started looking into it, and I'm looking at these companies, and I'm looking turnaround time 2 weeks. I'm like, 2 weeks? Geez, that's a lifetime for a late-stage cancer patient. And so I got really discouraged with that. And then, of course, the price tag, everything. And I thought, well, maybe I can just pay for the testing. I'll do that. I'll just pay to get him tested. But then I thought, well, but I didn't pay for my sister-in-law to get tested, and I didn't pay for this friend to get tested. Why would I pay for this one? And even if, he came back and had a target that could be treated, they had already racked up $100,000 in debt. If he were to use something off-label, not covered by insurance, can they afford it? And so I got so discouraged about this, and especially when I thought about, what can I do about it? And short of wanting to pay for everybody's cancer test, I thought, this really, really drives home why I decided to educate people, why I decided to shift my career from research to education, because I really think that linking patients to these cutting-edge technologies is really important. And I think what's standing in the way there a lot of times is education. It's educated patients to know that it's available to ask for it. It's educated healthcare providers to know that it's available to offer. It's educated stakeholders, whether it's the FDA or whether it's some manufacturer of some genotyping chips or whatever. There are stakeholders everywhere that need to be educated in this space. So that anecdote, that really drove home my mission and the importance of education. And that's one of the reasons that I was really excited to come here today to share what I know about this field with you, and really, just thrilled that the organizers chose this topic for the conference this year. Thank you very much.